Okay, I just got a message here that I need to click on 1 second. Okay, so 240 million plus sequences in UniProt. So these are all protein sequences. And that amount is growing and growing because of like studies of microbiomes and everything. There's lots of new organisms coming up, lots of new proteins as well. And if you look at the structures from the PDB, then only about 2 million or maybe 3 by now of these sequences can actually be mapped to PDB structures. Yeah, so for most of these sequences, we don't actually know the structure or the behavior. And part of that could be, of course, that these proteins do not have a very particular fold, or the fold has not been determined yet. And this has also been coined the dark proteome. Now, I was using this slide until recently, but of course, now with AlphaFold, a lot of this can also be addressed directly. So the question then becomes not so much, okay, is the protein, does it have a fold, but like what other types of behavior might it have? Because AlphaFold is clearly not perfect in predicting structures. And this is an example here where you can see that for lots of regions of a protein, it can't find any solution. So what is happening to those regions? That's still a question, a very open question. Now, another thing to keep in mind as well is that within proteins, you have, they operate within an in vivo environment, so in the cell. And this is a picture that sort of illustrates that. Lots of molecules densely packed to each other, constantly interacting, metabolites present, ions, of course, water. There's a lot going on inside a cell. And this is also, this is also important, but very difficult to capture in current methodology. So when I say, okay, you've got protein sequence and structure, there's lots of ifs and buts there as well. On the one hand, on the sequence side, you might have conserved sequence position and the tyrosine in the middle here. But local interactions also play a role. It's possible, for example, that the lysine interacts with the tyrosine side chain in order to change its behavior or what the tyrosine is capable of, because it might be forming interactions with the aromatic ring, for example. Whereas if this is a methionine, this could be different, and that tyrosine then can also function in a different way within the protein setting. There's, of course, also interactions between amino acids that can make a difference in the way the secondary structure or the dynamics and so forth. So local interactions are very important, but when you look at just conservation of one column, this is not taken into account. Another thing that happens to proteins a lot is post-translation modifications. So this tyrosine could be phosphorylated, for example, which will also very much change its characteristics. So there's a lot of stuff going on the sequence side that we don't know very much about. On the structure side as well, it's possible that a protein might adopt different conformations. It could go from a fully folded state to a partially folded state to completely disordered, and it might move between these different conformations. The typical view, I would say, is a defined static view of proteins where it does have one defined fold, but of course, there's also a probabilistic dynamic view of proteins. So looking at them from the perspective of, okay, they're capable of adopting very different conformations and different behaviors. What might then happen in the particular context of the cell to the protein might depend on the environment. For example, only partially folded if it's in contact with a membrane, and only fully folded when it sees the right binding partner. So the environment is very important as well to determine how the protein behaves. So the context is important in any case, and this is not something that is easily captured. Another way to visualize this is if you look at an example energy landscape of a protein here, the lower the energy, the better. In principle, what AlphaFold does is predict the one low energy conformation if it's available of a protein, and it is very good at that. And this already really opens up the whole field of structural bioinformatics. But of course, it's possible that this protein might also adopt different conformations, and move between them. and that some of these energy wells might suddenly become a lot lower when it is in the right environment. And this is not something that AlphaFold is really capturing at the moment, because also there's not many data available for this. So it's hard to train machine learning methods on that. So you could say there's sort of a void in between this sequence and structure information about the possibilities of protein behavior, like what is the protein capable of? And this is more or less what we're trying to address in our lab. So if you then take a step back, what is always important in proteins are the local interactions. So amino acids that are close to each other in the sequence are covalently bound. They always will interact at one point or other in the life of the protein. It might partially unfold. The local amino acids are always close to each other and they will interact. And this is something that was already pointed out many years ago by Shoshana and Marianne Roman. That's 30 years ago now. That this is a very important feature of proteins, and this is really what we're focusing on in our research. Now, I'm going to explain a bit about the methodology that we've developed and how we then use this in this context. Basically, what we're using is data from NMR, so NMR chemical shifts. These chemical shifts are basically measurements that you record with NMR for the protein in solution. So it is adopting, if it is adopting multiple shapes or dynamics, these NMR chemical shifts capture that information to a certain extent. This is also available from a public data bank, the Biomac Bank. So what we did with these chemical shifts, and I will explain this in a bit more detail later, we made estimations essentially of parameters like backbone dynamics, side chain dynamics, and secondary structure population by methods developed in other groups and also together with Michele in Cambridge. And we got a data set for 2,000 proteins, just over 2,000 proteins, 220,000 residues about, with which we made a predictor essentially to predict the backbone dynamics from single sequence. Yeah, and I'm going to explain in a little bit more detail how we got to this and how that's relevant for what I'm going to describe next. So first of all, in computational methodology development, what is very important is your data set. If you don't have a good data set, you might have the best AI methods available. They won't work. AlphaFold only works because of careful curation of UniProt and the protein data bank over many, many decades and all the work that scientists have put into this. So we started from the Biomac Bank, where you have deposited NMR chemical shift data available, and then made sure that we only took chemical shifts recorded for proteins in a particular set of conditions, so between pH 5-7, a relatively narrow temperature range, no order or disorder inducing agents in the sample, like for example urea. Made sure we had lots of chemical shifts for H, C, and N atoms, and then also we applied correction methods like chemical shift rereferencing to get this curated set. So this is already very important, and often in computational methods, this is what we spend most of the time on, I would say, is just getting the data right. Once you have good data, then there's all kinds of things you can do with them. So clean and dependable data is essential. Now, what we then did is we used this curated chemical shift data set, like I said, to run random coil index and the other methods that I mentioned to get an estimation in this case of the backbone dynamics. So how much does the backbone of your residue move? But then we have this information for 220,000 residues, which is quite a lot. So basically what you do is you try and relate the information for the protein in solution directly to its sequence. Now, this is less accurate, of course, than doing very specific NMR experiments, but in this case, you have lots of data. Also, this incorporates movements up to the low millisecond time scale. So basically what you have is a data set that relates the roughly accurate behavior of the residues to the sequence. What we then also first did in this, and this is also important, is that you validate your estimation. So look at, okay, does this data make sense in light of other things that we know about proteins or amino acids? So in this case, we plotted, for example, here you have the value that we got. So where this high values here mean that the amino acid is very rigid, low values mean it's very dynamic. And then if you look at residues that are typically order inducing, where 0 basically means that this amino acid has no preference, yeah, and positive values mean it does have a preference for this kind of states. Lower values mean it does not like this state. Then you can see for order inducing ones that, yeah, they really like to be very rigid, which is normal. I mean, we're talking about amino acids like valine, isoleucine, yeah. If they are neutral, like asparagine, arginine, and such, alanine, then yeah, you get a flat curve, so they don't prefer any particular rigid or flexible state. And the typical disorder-inducing ones, yeah, they clearly have a preference for movement. So this all makes sense. We also correlated to actual recorded NMR S2 values in this case, yeah, where you also get a correlation, which isn't fantastic, but still quite reasonable. So it means that the data that we got out is usable and useful. We then developed this predictor. Again, I'm not going to get into the details here, where essentially, so Dynamine, where you have a sequence, you run this prediction, and then you get a profile for your sequence based on the machine learning, where, yeah, it's very quick. Where this is your sequence, this is your prediction. High values mean rigid, low values mean flexible. Yeah, that was developed with Elixir Cilia almost 10 years ago as well now. Yeah, the web server is available. You can run predictions with this. And to give an example of what that then means, because that's also important to try and gain an understanding of what the information that your predictions carry. We looked at, this is an example for the human crab binding protein. So these are the predicted scores. Here you've got the sequence, so it's a long protein, 2,442 residues, predicted backbone dynamics. And this is what we knew at the time about this protein. So here in dark gray are folded domains, and these two different colors here are domains that fold upon binding, or regions of the protein that fold upon binding. If you look at the folded domains, you can immediately see that these are higher values. Yeah, that makes sense because these are regions that want to fold. For the two folding upon binding domains, yeah, the values are lower, but there's still peaks in the whole profile. If you plot all this information in a histogram where these are high values, so this corresponds to one here, is high here, and the 0.4 is low. So this is rigid, this is flexible. The residues in structure domains tend to have very high values. The residues in the folding upon binding are here, and all the other ones, which are basically disordered linker regions, have very low values. And you can see that there's like a region between 0.7, 0.8, where you have a mix between the two. Yeah. And at the time, what we coined this to be context dependent. So essentially, if you're above 0.8, then you are very likely to have a structure. If you're below 0.69 in this case, you're very likely to have flexible region in your protein. But if it's between these two, then it could be either, and then it will be very context dependent on, so depend on the environment. So this was a first step towards trying to define this, where your protein might have different behaviors depending on the situation. Now we did the same thing, exactly the same methodology development, but for other flavors of this prediction. So we have the backbone I told you about already, that's Dynamine. We also did sidechain dynamics, and then also helix, sheet, and coil propensity, essentially. But again, all derived. So this is not helix, sheet, coil in the sense that if I have the sequence, this will form a helix. This is helix, sheet, coil in the sense that this has a preference for helix, sheet, or coil as observed from the NMR data. So once you have a prediction like the predictor like this, at least, then you run it on many different sequences, and you think about it, and you look at examples, and you think about it, and then that made us really define what these predictions mean and where they come from. This is also important. Which data do you use, and then what do you see in the predictions in the end? So this came from directly from experimental data. So it represents to some extent the actual behavior of a protein solution. If there's loops flapping about or there's partial unfolding, the chemical shifts will capture this somehow. It also, and this is a very important point, covers the whole range of proteins. So you have not only folded proteins, you also have IDPs in the data set because you can record chemical shifts for those. So it covers a wider range of protein behavior than what you get if you only use the PDB essentially. The third thing, which is more methodological, is based on linear regression. So you extract overall statistics, so global trends essentially in the data. So in a way, what this method does, because it is quite looking at overall statistics, it's looking at it's capturing different behaviors of proteins in solution, and it covers many different kinds of proteins. You sort of predict the protein's average behavior in average context almost. So it's a very statistical view. What is your sequence capable of, more than what is it exactly going to do? You could also call this emergent property. So things that are inherent in the sequence, but that, you know, might or might not happen depending on the case. So to go back to the local interactions that I was talking about, one place where local interactions are all determining is in early folding, because if you have a protein that folds and it's completely unfolded, it's only going to be the local interactions that really are going to be there in the beginning, because there is no context, there is no fold of your protein yet. So essentially in early folding, yeah, you form these local contacts between residues close to each other in the sequence that form like local structural elements, which then help the rest of the protein fold. That's the idea. Some of these conformations provide enough context for folding to proceed. So we thought, well, this is a good test case, because our predictions should be able to really capture elements of early folding essentially in sequences, because that's what we are doing. So what we did is develop a database called Start to Fold. I'm not going to get into the details. This was with Michi and Rita here down the corridor, using NMR and mass spec hydrogen deuterium exchange folding data from literature. So Rita especially went through literature to pull out all the information. We classified it and then selected an early class from NMR post-labeling experiments. So basically, these are residues that, if you have a protein that folds, you unfold it, you restart the folding, and this is where the folding really starts. This is one of the sequences that we had in the database. This is whale, apo, myoglobin. This is so it's a very well studied protein. Here you've got the sequence, in black you've got the backbone prediction, and in green you see where the early folding residues were determined by completely independent hydrogen deuterium exchange methods, and you can already see that these regions correspond to peaks in our profiles, which is of course very satisfying. But that's not enough. That's just one case. We also did a whole analysis looking at how these things are related to each other. Again, I'm not going to get into that, but it was enough to convince us that, okay, we can predict this early folding for protein sequences. And what we did then was use as input features not the actual amino acid sequence, but the biophysical features that we predict from the sequence. So backbone dynamics, side chain dynamics, and then also secondary structure propensity. I used a 5 residue window, 25-dimensional vector. In this case, we used a support vector machine, I think, for prediction. So, and this was on a very, I hear someone talking, but I'm not sure. Okay, so this is on a set of 30 proteins, so that's very few in terms of machine learning, but it was enough to train a predictor that essentially says from a protein sequence, okay, here, the sequence seems to be able to form enough local interactions that it can fold. I'm, again, not going to get into the statistics here of how well it performs, but it had an area under the ROC curve of like above 0.8, which is not bad for this kind of approach. And the Matthews correlation coefficient of about 3.5, I think, on it, 0.35 on average, which is on par with, like, you know, secondary structure prediction, like say 15, 20 years ago. That was done by Daniela Raimondi in the lab. Again, we did independent validation. I will go over this part rather quickly, but you can see that if you look at the early folding regions, these are the ones where we predict early folding. So this is data from mass spec, so again, independent, not in our training set. where we predict early folding. Yeah, here you get much higher values than where there is no early folding, also according to the mass spec. Whereas when in regions that have intermediate folding, there is no difference in our prediction. So that means that it seems to capture also based on mass spec data, the early folding. In this case, this was for apomaltose binding protein data. Another case study is myoglobin here. So these helices A, B, G, and H that are related to the structure here were shown to be the ones that fold the first. Whereas for leg hemoglobin, same structure, different folding pattern that has been determined. You can see that the profile that we predicted is also already very different. And here indeed, it was these three helices that were shown to start the folding process. So it is capturing something here as well, based on, again, completely independent data. We looked at many other things, like, for example, aggregation. You can see that for residues that are amyloid forming, which are in light pink here, for some residue types like tryptophan, there is no difference in what we predict in early folding, but for some like leucine, amyloid forming or leucines that are in regions of the protein that form amyloids have a much higher early folding propensity, which makes sense because these are things that happen when the fold is not completed yet. So these are all, again, this is circumstantial evidence, if you want, that the method works. But so this, in any case, gave us the idea that we can use this information to get a different perspective on the protein, essentially, from what you get from sequence or structure. We also developed a disorder predictor, which was recently published this summer, also that was evaluated in the independent KITE competition, where it scored quite well. Also an aggregation one that's been published, for which we also developed a separate database. So basically what you get is this emergent picture of proteins and that captures their possible behavior. And then you can start to do more things with it. So here's an example, for example, for a tin barrel, where here you have a sequence of the tin barrel. This is the structure. This is the secondary structure related to the sequence. And you can run predictions. This is backbone dynamics, and you can see there's like quite a lot of variation, and it relates to the secondary structure elements. But what you can do now is use evolutionary information. So this is a structure-based alignment for tin barrels. You can then run the prediction and plot these all mapped on the multiple sequence alignment. And then you can see that there are persistent peaks in this profile and also persistent troughs, that you can plot as in a box plot kind of way, where in black you have the median of all the values for that column in the multiple sequence alignments, all the predicted values. And then you have a blue range, which is the first, third quartile. If it's dark blue, it means that this sequence is very conserved. If it's light blue, it means that the sequence is very variable in terms of amino acids, right? So not in terms of the predicted values. So what you can see is that there's regions that are very conserved and that also have almost no variation in the predicted characteristics, like here. Makes sense, of course. But you also have regions that are quite conserved that actually have quite a lot of variation in the predicted biophysical characteristics, or in this case, the backbone dynamics, which indicates that maybe there are other things important for that region. You also have regions that are very variable, but in fact have quite conserved biophysical profiles here. So there is a very bad correlation between the amino acid conservation and the conservation of these predicted characteristics, which is interesting in itself. You can look at, how does it relate to helices, for example, and to the beta strands. You can also do it for other predictions, like in this case, this is a beta sheet propensity. And this is also interesting because here you can see that some regions have quite a lot of sequence variability, but have a consistent beta sheet propensity peak, that is conserved essentially. Here's other ones, but then you also have here a conserved region that forms a beta strand, is very conserved, but in fact does not have a very strong beta strand propensity, just like here in the beginning, and this one in the middle. So what that means, we don't know, but it's likely to do with something that has to be present in your sequence in order for this protein to fold correctly. And then there's other ones that I'm not going to get into, but essentially, so what we're trying to do now is get a sense of what might be in this void by using these biophysical predictions to try and relate the behavior of proteins to things that we can observe, like folding and so forth. And this is not something that you can directly get from evolutionary information, nor from the final models in structural biology. Of course, you could do NMR experiments and everything, but that's when I say structural biology, I mean the final folded structures. So the concept then that we have now is that you can replace amino acids by their biophysics. So say you have a sequence, you've got that tyrosine that I mentioned in the beginning. You basically replace that by a multidimensional vector of these biophysical annotations, things like backbone dynamics, early folding, and a local sequence-dependent biophysical signature. But then also you could include things like charge or other traditional attributes of amino acids. And then you can use that framework for things like alignment, homology detection, and protein design. That's the idea. We tested that was Gabriele in the lab a while back. So basically, we tried to align sequences that were homologue sequences that were evolutionarily very far removed from each other. And if you look at traditional sequence alignment methods here, the performances were not very good. Our method that was using not the sequence, but the biophysical profiles. So it was aligning, if you want, the biophysics of the protein in multiple dimensions, called Rigapolo. We got much better performances on the alignment. I mean, they still were not very good. So this is residues that were exactly correctly aligned, shifted by one residue and shifted by two residues, but still it was doing much better than the traditional sequence-based methods, because in essence, what the concept there is that what a protein conserves is its biophysics, not its sequence. Only if amino acids have to be in particular places for things like enzymatic function, then yes. But in general, evolution doesn't really care as long as your protein works. So this is what we think this is picking up. We developed other methods like warp for very quick single sequence global homology searches, and then also for identifying proteins with the same structure from sequence only. And this has now come into this whole framework that we have available, the B2B tools, which was published last year. So you have a website where you can enter sequence or sequences or multiple sequence alignments, and you get these profiles back. So all the different predictions that I was talking about. You can also get, if you enter a multiple sequence alignment, this biophysical conservation that I was talking about. There's also JSON files that you get where you can download all this information. We also, as Shoshana mentioned, we developed a website for exploration of SARS-CoV-2 proteins during the lockdown to have a joint lab project that we all worked on together. That was also in collaboration with Anton Veenstra at the Vrije Universiteit in Amsterdam, who provided epitope predictions. So where is this protein likely to bind? So this is also a website that we developed that's available online, where you, again, also get this biophysical conservation concept. Also something that we've been working on for a long time is, like I said, what's also important is the context of the protein. And one thing that is possible to get, or data that is reasonably reliable to get these days, is post-normalization modifications from mass spectrometry proteomics data, essentially. And this is done with Padmanabhan Ramasamy, who is a joint PhD student between my lab and the Compomics lab of Lennart Martens at Ghent. And together we developed this COP3P sites, which basically gives you, for any human protein in this case, the phosphorylation pattern and how reliable this is. So I refer you to the website for this. But essentially you get for a sequence, you can see where you have phosphorylation, how reliable the phosphorylation is. So is it observed in multiple experiments that are done by different people? You can also see them mapped onto the structure. You can also click to AlphaFold. So it's a very nice website. So if you're interested in if you're working on a protein, you're interested to see what's happening in terms of phosphorylation, this is the place to go. But what we also did is link this to the biophysical characteristics that I was talking about. And then interesting patterns come up. See, and this is not published yet, but we submitted it and it's now in revision. What you can do then is look at phosphorylation sites and see, okay, are there any patterns in these biophysical characteristics? And here you see the distribution for backbone rigidity. So the dynamine I talked about, where 1 is rigid, 0.4 is flexible. For phosphorylation and non-phosphorylated sites, where we made sure that we basically extracted data so that the non-phosphorylated set is very similar to the phosphorylated set, except that there is no evidence for phosphorylation. And you can see that typically phosphorylation happens in much more flexible areas, like right at the edge of the bottom edge of the context-dependent region that I was talking about. Whereas for non-phosphorylated sites, you are at the edge of the rigid region. Same for early folding, there's also clear difference. So the early folding signals for phosphorylated sites are much lower than the ones for non-phosphorylated sites. And disorder propensity, the same picture. So here, the sites that get phosphorylated tend to be in much more disordered regions. So this is interesting information. This is of course very large scale data over many, many thousands of proteins, but it does give an insight into the physical factor, the biophysical factors that have to be there in order for a protein to be phosphorylated at a very large scale. And this can then be translated back into predictors, of course, to more reliably predict, for example, phosphorylation sites. And we're doing the same thing for other post-translation modifications as well. Another thing you can do once you have all these machine learning models is do interpretation. And this is with the AI lab here at the VUB. We developed this method where basically we train the black box, so the eFold mine that I talked about. What you do then is you run it on many thousands, hundreds of thousands of sequences, and then you basically train a method on those sequences that you can interpret. So the method we developed, you cannot interpret, it's a black box. You cannot extract any physical meaning from it, but some machine learning methods you can. So you can then run those methods on the predicted data to see what the black box predictor was thinking essentially. So this is the idea. We have a website for this as well, where you can explore all the different 5 residue early folding fragments and the relationships in terms of amino acids to each other. And essentially what we found out from that is, so if you have any fragment of 5 amino acids where the middle residue is early folding, basically what you see is typically is that the residue and the subsequent residue have higher backbone rigidity. They also have higher sheet propensity, and the residue at position -2 has higher helix propensity. Now we try to validate this somewhat in case studies, but we don't know exactly what this means. But this is a signal that is present in the prediction, so it probably does mean something in terms of what your sequence has to be able to do locally in order to form local hydrogen bonds that then progress into a fully folded secondary structural element. So that's the website. We've also done other analysis, E. coli cytoplasmome and secretome, together with Tassos Economy and Leuven, who did all the experiments, and then we did the interpretation from the biophysical side. So here you can see that proteins that stay in the cytoplasm of E. coli and ones that get secreted. Here, this is the early folding. Yeah, they have the same number of early fold-ons, if you want, per 100 residues, but the ones that get secreted tend to be much shorter than the ones that are cytoplasmic, and also they tend to have lower values. So there's additional analysis in the paper that are definitely interesting to look at. But essentially, what this shows is that proteins that have to be secreted need to stay reasonably unfolded and cannot immediately fold, otherwise they will just not be able to exit the cell. This is also, we're continuing this, collaboration by looking at 2 proteins that one exactly the same structure, one is secreted, one cytoplasmic, to see whether you can change their biophysical folding behavior. And this is also now a paper that's in revision from Tassos group, where we have contributed. Another paper that Shoshana also mentioned was the secretability of protein fragments in yeast, where basically they made random fragments of proteins, expressed them in yeast libraries, and then saw whether those were secreted or not by the yeast. Experimentally, I'm not going to get into the details, but what we observed again is that fragments that were enriched, so these are fragments that were secreted by the yeast, and depleted are fragments that were not secreted by the yeast. You can see again here that the backbone rigidity tends to be much lower in exactly in the context-dependent region, in fact, for the enriched ones, whereas they were much more rigid for the depleted. Same for disordered content. So there also the enriched ones were much more likely to contain disorder than the depleted ones. So again, showing that proteins need to have ambiguous behavior, even if they have exactly the same folds in the end, they need to be more ambiguous and flexible in behavior in order to have certain other behaviors like the secretability. Yeah, also in secondary structure, there were signals in relation to alpha helical. Yeah, so typically there was more alpha helical propensity in the depleted signals versus the enriched. For beta sheet, there was not much of a difference. And also, of course, there was more random coil propensity in the enriched fragments, so the ones that were secreted. Also for protein design, and this is something that we really want to look into now, is can we, in fact, because we can, to a certain extent, help with identifying where protein folding might start, then can we also help with protein design by essentially using this representation to capture features of proteins that are not evident from the sequence nor from the structure, which might also help in designing more natural proteins that have dynamic behavior, essentially. And this, we have already done some work in, with people at the Université de Liège on that, where we could show that there is definitely some signal there that is useful, but this is not consolidated yet. So with that, I will refer you to our website for more information on all these tools and the work that we've done, and then acknowledgements. So there's many, but we have a nice group at the Bio2Bio group now with people that have contributed to this. These are people that I've mentioned collaborations with, and all the people that formerly were involved and have contributed to this work. And the funding agencies, of course. And then I will stop with questions. And thank you for your attention.
Thank you very much, Wim. So open for question now. Please don't feel shy and ask questions, please. Okay. If no one comes up, in the next half a second, I have one. Did you use the biophysical annotations to align disordered regions, sequences of disordered regions?
This would be helpful. This is something we're working on. It's tricky because what is the ground truth, right? I mean, it's hard to align disordered regions, so you don't really know when it's right and when it's not right. But what we are trying to do now is to come up more with a way of so are there local signals within disordered regions that have a particular biophysical profile, if you want? Mm-hmm. And then using this approach, again, using multidimensional space and some scoring system, you could run local alignment methods essentially to pick up similarities between different disordered regions, which I think is more relevant in relation to disorder than trying to align, global sequences, just like with the motifs essentially that one can see. Yeah, it's possible that there are features.
You can combine this with the motifs and
exactly. So on. Yes, indeed.
I have a question for you here. Is there units? Oh, okay, now it jumped. Is there units using for the measure of those dynamics, or is it just kind of a probability? So this is the question. What is on your scale?
Yeah, no, this is a good question. So random coil index extracts a value, I think between 0 and 0.55 or something, that they calibrated against some molecular dynamic simulations, which was then transformed into an NMR, a measure that's used often in NMR, which is the S2 order parameter, which essentially is the orientation of the NH bonds in the backbone in relation to the molecular frame. If that value is 1, it means that your NH bond is completely fixed with respect to the rest of your molecule. If it's 0, it means it's completely random. So this is the value that we used for training. So 1 means fully fixed, 0 means completely random, essentially. But whether it actually has that meaning, I'm not so sure. A matter of debate. I mean, this is what we said in the original paper, but I think it's a bit vaguer than that. No.
You're allowed to be vaguer. Biology is complex enough. I have another question here. What limitations did you have while performing NMR regarding bigger proteins? From what I know, protein size is a big drag problem.
Yeah.
But in NMR, how does it deals with bigger sequences? So
yeah, also a good point. There are no bigger sequences in our data set. This is clear. But the idea is that because we are predicting essentially local sequence characteristics, right? That are, of course, there's influence from, if you have a big protein, you will have more buried residues essentially compared to small ones. But the idea is that the essential local characteristics of your sequence are not going to be any different in a big protein compared to a small one. And there are enough buried residues in NMR structures that this is not a problem. I mean, this is hard to quantify, I would say, but I don't expect that there is any real problems with that. We did look into it to a certain extent, but like, for example, the example I showed with the early folding there, from the mass spec data, which is a big protein, it works fine, the prediction on that. So that's sort of circumstantial evidence, I guess, that the predictions are working well for those as well. But they were not in the training data, long proteins, no.
So another question from the same person, I think. Also, could conformational domains be phosphorylated as long as the context allows that the regions undergoes unfolding?
Yes, so very good point. This is also something we're looking into. There are phosphorylation and other post-translation modification signals in regions that, according to either experimental protein structure models or AlphaFold generated ones, are completely buried, which would indicate, of course, that there is conformational ambiguity. So we're trying to use that data to try and predict where your structures, in fact, might be, where there might be a story, essentially. So this is, I think, that's going to be extremely useful data, because you get it from in vivo experiments that show what's actually happening to your proteins in the cell, and then you can translate that back to what we already know for individual proteins from structural biology. And I think that would be a gold mine in terms of data and understanding how proteins behave in the cell.
Yeah, concerning phosphorylation, I wonder, did you integrate or did you map data on conservation of phosphosite regions and on the abundance of the proteins in the cell? Because it's well known that some phosphorylation sites are kind of haphazard and others are functional. So did you try to do this data integration?
We did all these analyses, and this is what Bob Manaman has been doing also over the last year. But essentially, we do not report, I think, a phosphorylation signal unless it occurs in, I mean, if it only occurs in one experiment, then it's meaningless. But of course, if you see, say you have 10 experiments and it occurs in 5 or 6 of those, then it is going to mean, it means something. Also, so a range of proteomics experiments was used, like some where the phosphorylation was, forced in a way, but then also ones where it was not, that had nothing to do with it. So there's a balance between the kind of data sets that were used as well. So the information is very reliable, I think, and also being mapped back now to uniprots, because a lot of the, it confirms some of the predicted sites from uniprot, but it also comes up with lots of new ones.
Yeah, but what I was referring to, it can be completely reliable, but not functional.
Yes, absolutely. Yeah, this you never know. But
yeah,
I mean,
the sites are conserved, you have more, and apparently there was a paper out that, low abundant proteins, when you look at the phosphorylation site, those which are more highly conserved in low abundant proteins are functionally, selected for something like that. So there's a very nice paper that shows that. It would be wonderful to have these
in the recent analysis of that Maneman did, we have all these relationships between abundance and phosphorylation, but I cannot remember exactly what the conclusion is there.
It's definitely work in progress, because all this is really difficult to interpret. I have another question here. Can we expect for a computation of the biophysics dynamics of proteins during an enzymatic reaction or any interaction between proteins and its ligands?
Yeah, so we never actually really looked at enzymatic activity or catalytic sites as such, whether there's a signal there. I don't know. This is, I think, yeah, there would be some biophysical conservation, I think, but I don't know how that relates, and I think that really depends on the enzyme as well. There, some I think are more rigid than others, but I think probably even Vercees, if he's still online, is a better person to answer that one.
Yeah. Wouldn't you need, the actual chemical shifts on the folded protein to see what's going on, like encounter complexes between your ligand and the protein, and link that to your dynamic properties?
Ah, you mean the change here? Okay, so I'm not sure if I understood the question. Yeah, no, we can't predict the change. So the idea is that we can predict whether it's capable of change, though. That's
right. Yeah, but you could relate this whether it, in some systems, not on a genome scale, of course.
But to use this in individual cases, you always need to use as much information as possible, and I think this adds an angle to that, also in terms of how to think about it. That is useful, especially if you use the evolutionary conservation. It can help you understand what your protein might be doing, but by itself, I don't think it can do much except like detect things like homology and everything, because of this biophysical signal that's present. Yeah.
Any other questions? Please do.
I have a question.
Yeah, Anastasia.
Thank you. That was a very nice presentation, very clear. I think everyone enjoyed you, especially the first part where you very clearly explained what is important to train the model. So I was wondering if you try to kind of look back to the local structure of the protein, because when in one of your last slides you showed that you were trying to predict on the windows of 5 residues, the properties of like per residue property. I thought, I mean, have you maybe looked at whether you were capturing some specific 3-dimensional motifs that might be enriched in early folders?
Yeah, no, that's a good point. We actually did look very extensively at one point in the relationship between these predictions and what the structure is like, but this did not really go anywhere, and I'm still not 100% sure why. But yeah, so just to give you an idea, what you often see in like a helix, for example, is that there's like a big helical propensity signal at the beginning and the end, but there's like a beta sheet signal the other way around, a beta sheet signal at the beginning and end, but a helix signal in the middle, for example. I don't know what that means, but it's something that I've seen multiple times. So probably this means that it imposes some kind of behavior on that helix. For the early folding itself, this could be in terms of what actually, so in the structure, this could be a beta strand, could be a helix, could be a loop. there is no real connection between the two in that sense. So the idea would be that if your local sequence can form, a local hydrogen bond, which seems to be the signal that you have like beta strand, so some sort of extended propensity and rigid, so that part stays present, and then I guess helix maybe to turn around and make a hydrogen bond. This seems to be the pattern that comes out, which is quite interesting. But we would have to do many experiments and everything, which we don't do. But if someone's interested, I'm happy to work with them.
Thank you.
I had another question. Sorry, someone else wants to ask something?
Well, I had the last question, but
Yeah, go ahead.
there's
Go ahead.
many. Yeah, it's fine. I can always ask him later.
No, you go ahead, Anastasia. We have time. We said, 20 minutes questions, so we have time.
Okay, because I see some questions in the chat as well.
Ah, yeah, I see also. Yeah, okay. So go ahead, ask yours, and then then I'll take up the other question.
So I was wondering because so you showed that PPI work with Tassos. I mean, I was also briefly involved in that. So I was actually surprised because they really focused on the three-dimensional frustration in the protein rather than frustration in the secondary structure, which are, I mean, like you, I would probably first think about. So I was wondering, what is your take on that?
Yeah, I mean, we've been looking at this for many years now. In fact, I think we started about 5 years ago, 6 years ago, I can't remember. And in relation to PPI A and B, the predictions that we had for early folding and the residues that you had to change in order to get from A to B, if you want. So, you can run this very quick, change the sequence, rerun predictions, and see where the biggest changes are, essentially, to make A go to B. We identified, just purely based on the sequence, the residues that were the most important, that they then afterwards also identified from structure, essentially. But I think, in this particular, the last paper, I think they just used the frustration method because, the structure is always going to give you lots of information about what happens in the end, of course, I think.
So you mean the tertiary structure frustration could also be detected from?
Yeah, I mean, the early folding is only the beginning. Okay. It is very important, but it's not the only story, of course. You might start off right and then not get anywhere because the rest of your, you don't get the right interactions between the different elements that get formed. I mean, there's a lot more to it than just the early folding. Again, it's the predictions that we have are very local and sequence-based, which is very relevant, but of course, it's not the whole story, because in a folded protein, as you say, there's a lot of context, right? I mean, if a helix is formed, then suddenly this completely changes the environment of residues that are close to it. And then the local interactions are still important, but also the fact that helix is there. So, I mean, folded proteins have an enormous amount of context in that sense for other amino acids that are not yet folded. So it's
And there is also
a different perspective, a different stage of the
And there is also data on non-native local structures in unfolded proteins that are actually, are not what is at the end incorporated into the structure.
Mm-hmm. Yeah,
there's quite a bit of data on that.
I think that's what you also see in these biophysical signals. That's like sometimes you have like I said, a helix that has consistent beta strand propensity in all proteins evolutionarily, or beta strands that have like, a bit of helix propensity. That's less the case, I would say, but in helices, definitely there's an enormous amount of variation, which is conserved. So it must mean something. Also in transmembrane helices, you often see like very consistent signals that are like, I don't know what they mean. But
you need alternative pathways, somehow. Anyway, this was, at the time, a big discussion in the folding community. You have non-native, structures, local structures, and so on. There is another question.
But to just, if I can.
Go ahead.
Right? So the study with the people in Liège, they wanted to design a tin barrel, but they got a completely different fold, like a Rossmann fold, I think.
Mm-hmm. And it's not too far.
Not too far, but not the same. But we could determine essentially from our biophysical profile, we could distinguish between the fold being either Rossmann or tin, because the they all want to form the same secondary structure, but it's the order in which it forms that is important. And that came out of that, just purely looking at these signals, which I think is, because it's the progression of, so again, the emergent properties, if you can call it that.
Okay. Now, Tommy Lazar is asking a question. Can you comment on the effect of the environment in the various subcellular localizations on the biophysics of the protein?
Yeah, good question, Tommy. Yeah, so we did actually look at that, and I even have a figure somewhere, and we do have a grant for that now, an FWO grant to look at how these things are related, and also how post-translation modifications play a role. But for example, I cannot remember off the top of my head, but I think in the nucleus, proteins tend to be a lot more flexible and have like more beta strand propensity. I think I do not remember, but there are definitely signals there. I can show you, seeing as you're just 2 doors down. Yeah, but there are definitely different, there are definitely different characteristics and of proteins in different subcellular locations. Yes. Yeah.
Another question here, can we expect one day a command line version? I'm not sure of what, but could be really useful in some pipeline. So
there's totally a command line version already available. Yes, absolutely. I didn't mention that, but it's in the paper. We have like, Jose and Adrian have developed also a full Docker package and integrating it into Galaxy and all these things so that you can basically run it on any sequence with the push of a button, and get it.
You are a good pupil of Elixir.
Do my best. Yes.
Good. We know where to go when we need. Any other question, guys? Yeah, we are done nearly. Good. Okay. Fantastic. Thank you so much. It was very, very interesting, and hope that you get other questions.
Yeah, thank you for giving me the opportunity, Shoshana.
Absolutely. I mean, this is only the start. We'll have, other stars from the university and the department also give talks at some point. Okay, thank you very much. Thank you to everybody, and
everyone.
Take care.
Listening.
Have a good week, and thank you, Wim, again for the wonderful seminar. Thanks.